Welcome, everybody, and thanks for joining again to the seminars of the EBD. This time we're having our seminar online with Anna Novoa joining also the EBD remotely. Today's speaker is Anna Novoa, as I said, originally from Sorry, I had the audio in YouTube on. Okay, so we have today joining Anna Novoa, researcher on in invasion ecology, really looking to the ecology, introduction, management of invasive species. And yeah, originally from Galicia, where she also did her PhD. But now she's researching in the Czech Academy of Science in Bruchonice, although she recently got awarded by the Ramón y Cajal, and she's going to the estación in Almería. And yeah, however, today probably she's going to speak about her research that she did during her postdoc in South Africa, I guess. Just one note for the audience, please write your questions in the chat of YouTube. Don't worry, don't wait until the end of the talk. You can write your questions whenever you feel like, and then at the end, we will read them loud for Anna Novoa to answer. So that's all for me. Anna Novoa, thanks again for joining us, and the floor is yours.
Thank you. So good morning or afternoon, everyone. First of all, I would like also to say a big thank you to all of you for having me here today. A special thanks to Montse Vila for her kind invitation, to Maria and Javi for the nice and smooth organization of the seminar. Although I just heard it's the first one, so sorry for the first one online, so sorry for that. And thank you all for being here today. So during this seminar, I'm going to be talking about some of the work that I have been doing over the last couple of years, studying plant invasions in Kruger National Park. Some of it I studied during my postdoc in South Africa, and some of it I still do it now, funded by with Czech funding. So yeah. So first of all, I believe most of you, if not all, are already familiar with the term invasive plants, but just in case, I will describe it briefly. So due to globalization, plant species are increasingly being moved around the world from one part to another. And when a plant is introduced outside this native area, it might face a lot of challenges. On the one hand, the climatic conditions of the new area might be beneficial or not for its establishment. Also, the soils of the new area might also present good or bad conditions for the introduced plant in terms of, for example, nutrients or humidity. And also, unless it's introduced together with soil microorganisms present in the native range, which also happens, but in general, it will find a new soil microbial community, which will include mutualists, pathogens, or other types of species that might be beneficial or not for its establishment. Also, in the new area, it will establish positive, negative, or neutral interactions with other organisms, such as plants, insects, birds, mammals, or humans as well. And all of these interactions will once again facilitate or limit its establishment. Now, depending on all of these factors, the species will maybe not be able to establish, die, disappear from the new area. It might be able to establish and then maintain a controlled population in the introduced area, or it will be really happy and become invasive. And when this happens, these species, the invasive species, often have a wide variety of negative impacts in the introduced area. In fact, biological invasions are currently considered as one of the most serious threats to biodiversity globally. So it might have impacts on the soils, on the communities of other plants or native plants or insects or herbivores and so on. So now the Global Naturalized Alien Flora, or GLONAF project, has put together a global database on the occurrences of established alien plant species in the world. And this data is what you can see in this map over here at the top right of the screen or of the presentation. And there in this map, you can see that the hotspots of alien plant establishments are in the USA, in Europe, Japan, Australia, and New Zealand. But what you can also see in this map is that there are areas where there are not that many alien plants. And a clear example of this are African savannas. If we compare the map showing the distribution of African savannas on the top left with the GLONASS map on the top, on the bottom right, you can clearly see the mismatch. So savannas constitute the dominant vegetation type in Africa, covering around 60% of the continent. And until very recently, African savannas were thought to be less invaded by alien plants compared to other ecosystems. There were several reasons that have been proposed for this, such as the low rate of alien plant introductions in some of the regions, the adaptation of African savannas to fire, which might prevent the establishment of many alien plants, or the resistance of native plants to disturbance. However, this trend is now changing. And in the last decades, alien plants such as eucalypts or prosopis, Parthenium, or cacti have been increasingly being introduced, becoming invasive and threatening the conservation of savanna ecosystems in Africa. And despite this, little is known yet about the impacts of invasive plants in African savannas. So this is something that I have been interested in for the last couple of years, in which I have been studying the invasion and impact of alien plants in African savannas using specifically Kruger National Park as a case study. Kruger National Park is situated in South Africa, as you can see in the maps over there, and it covers an area of approximately 20,000 square kilometers of semi-arid savanna. Now, Kruger is one of the oldest and most widely known protected areas in the world. However, protecting such an area, almost 20,000 square kilometers, from the invasion of alien plants is definitely not an easy task. And nowadays, plant invasions are one of the biggest threats to the conservation of biodiversity in the park. There are 146 alien plant species that are already recorded as established. And here you can see some examples. On the top left, you can see the invasion of Opuntia stricta, the invasion of Datura stramonium on the top right, and then Parthenium invasions at the bottom. So you can see really thick infestations. Opuntia stricta in particular is one of only a few species that are currently considered as high impact transformer species in the park. The park refers to it as that, high impact transformer species. Opuntia is also one of the most studied and widespread species in the park, Opuntia stricta in particular. It's native from Central America, but it was first introduced to the park as an ornamental plant in 1953. It was introduced by staff members to plant them in the gardens, in the staff villages, but it soon became invasive. Now it's something that you cannot do anymore, introduce alien plants to your gardens. Now, soon after these first introductions, it became invasive, creating these horrible infestations that you can see in this picture. There were several hypotheses that have been explored to explain the invasion success of Opuntia stricta in the park. For example, in this paper that you can see over here, Foxcroft and colleagues studied the influence of dispersal pathways, fire frequency, management history, soil type, vegetation type, and water regimes in the reproduction and survival of these species. However, none of these factors appear to influence the invasion of Opuntia stricta in the park, with the only exception of a positive effect that they found of the presence of the leguminous tree Vachellia nilotica that you can see in this picture. So it seemed that the presence of Vachellia nilotica was facilitating the invasion of Opuntia stricta. Also, Foxcroft, this time with Regmanek, tested the hypothesis that baboons could be dispersing Opuntia stricta in the park. Baboons are known to eat the fruits and disperse both the seeds and fragments of Opuntia stricta. So if you take just one fragment of the plant and you put it on the soil, it can also root and become a new plant. So baboons are believed to disperse them. So Foxcroft and Regmanek thought that the distribution of Opuntia stricta in Kruger National Park could be linked to the activity of baboons. In the areas invaded by Opuntia stricta in Kruger National Park, there are mainly two native trees growing. The one I mentioned earlier, Vachellia nilotica, the two pictures on the left, and Spirostachys africana on the picture on the right. Now, Vachellia nilotica can grow up to 10 meters in height, more or less, and its branches are thin and they are easily breakable and they are full of spines. So it is not really suitable for baboon roosting. Baboons don't like to just sit and chill there because they fall and they get damaged with the spines. Now, on the other hand, Spirostachys africana also reaches normally 10 meters in height, but it has no spines, and its branches are much more stable, and therefore it is suitable for baboon roosting. You often see baboons chilling on these trees. So Foxcroft and Rechmanek hypothesized that Opuntia stricta plants will more frequently be found under Spirostachys africana than under Bacillia nilotica, because baboons will be eating the fruits and pulling the seeds and so on. However, in this study, they found the opposite. They found that Opuntia stricta patches were significantly more frequent under Bacillia nilotica trees. So they concluded that there must be some other explanation for the distribution of Opuntia stricta in these savanna ecosystems. So we decided to look in detail at the soil, because we thought maybe the soil is the key. Maybe the conditions under Bacillia trees are more suitable for the establishment of Opuntia stricta than the soils under Spirostachys africana. In fact, Bacillia nilotica was until really recently called Acacia nilotica, and acacias are well known to change the conditions of the soil. So we thought maybe the key is actually there. So we use the same study sites as Foxcroft et al. In the previously mentioned publications. And these study sites presents a typical savanna vegetation with open patches with no tree cover, and then a patchy distribution of trees. And there are two body species, the ones that I previously mentioned, Bacillia nilotica and Spirostachys africana, that make up more than 90% of all the trees in the study site. And also the individuals of these two trees, they are mixed in the landscape. Here you can see on the front right individual of Spirostachys africana, and at the end of the picture, and on the left, individuals of Bacillia nilotica. So in this study, what we decided was to compare soil characteristics such as pH, humidity, and nutrients, soil enzymatic activities as a proxy of the function of microbial communities, the diversity and composition of bacterial communities, and the percentage and speed of germination of Opuntia stricta between soils collected under the canopy of these two native trees and in open areas with no tree cover. And we also collected under these two trees and in open areas with the presence of Opuntia stricta. So we basically have presence and no presence of Opuntia stricta in each tree types under the canopy of the two native trees and in open areas with no tree cover. Now, with this data, we mainly aim to answer two questions. And the first of all, does Bacillia nilotica create these better conditions for the establishment of Opuntia stricta? So it will explain what Foxcroft and colleagues have been seeing before. And on the other hand, if Opuntia stricta is impacting the invaded soils. Now, first of all, I'm really sorry for this big table, but I'm going to walk you through it. So this table represents the results of two-way ANOVAs testing the influence of invasion on the one hand. So we have uninvaded areas on the left and invaded areas on the right, as well as tree cover. So soils under the canopy of Bacillia nilotica, under the canopy of Spirostachys africana, and in open areas with no tree cover. This is why it says none, sorry. And also their interactions on the pH, humidity, nutrients, and enzymatic activities of the collected soils. So just to clarify, the three studied enzymes, phosphatase, beta-glucosidase, and urease, play key roles in nutrient cycling. Phosphatase is involved in the release of phosphate from organic matter, beta-glucosidase is involved in carbon metabolism, and urease is involved in the cycle of nitrogen. So, although we found no differences in pH between any of these soils, what we found is that in uninvaded areas only, so just look at the green area of the table, humidity, nutrients, and phosphatase activities were higher under the canopies of native trees than in open patches with no tree cover. This confirms the null hypothesis that woody species in savannas create the so-called fertility islands. I see there is some construction going on. I don't know if you still hear me well.
Yes, everything is fine.
Okay, thank you. So as we can see in this picture over here, savannas are generally characterized by the presence of grasses and a patchy distribution of woody species, and generally really nutrient-poor soils. So we don't have many nutrients, and we have this scattered distribution of trees through the landscape. And due to their extensive root systems, trees and shrubs tend to extract moisture and nutrients from their surroundings, leading to an accumulation of soil nutrients, humidity, and microbial communities under their canopies. And this is what is called fertility islands. And this is what we are already seeing in our results. So these results were kind of expected, that the humidity, nutrient levels, and phosphatase activities will be higher under the canopy of the native trees. But now if we compare uninvaded and invaded areas, what is really interesting is that we also found higher humidity, nutrients, and phosphatase activity levels in invaded and in uninvaded areas, which is telling us that Opuntia stricta also creates fertility islands in invaded savannas. Another interesting result is that generally there were higher concentrations of soil nutrients and phosphatase activities under the canopy of Bacillia nilotica than under that of Spirostachys africana. So it's already hinting to us that there is something about Bacillia nilotica there. Now, if we look at the bacteria, here we can see the results of different diversity indexes. And the blue lines indicate uninvaded areas, while the red lines indicate the results for invaded areas. And then we see first the diversity of bacteria under Bacillia nilotica canopies, Spirostachys africana, and in open areas with no tree cover. So what we can see, once again, is that the presence of native trees increase the diversity of bacterial communities compared to open areas with no tree cover, where the lines always go down. But then if we compare these blue lines and the red lines, we can also see, once again, that all diversity metrics were significantly affected by invasion status, being higher in invaded than in uninvaded soils. And actually, invaded soils show the highest levels of bacterial diversity, which is quite interesting. And now here, forget about the small text. Just each word is just one bacterial OTU. And so what you can see is that on the left is the heat map, where the colors indicate the abundance of different bacteria of each of the bacteria there with a small font. From dark being low abundance to light colors being high abundance. So if we look at these colors on the right side of the heat map, which is indicated in green, you can see a higher abundance of bacteria under native trees, under the cover of native trees, than in open areas. Actually, the results for open areas are basically black. Only 3 of the bacteria that seems to be somehow abundant there. And now if we compare the right and left sides of this heat map, so invaded and uninvaded areas, once again, we can observe the higher abundance of bacteria in invaded and in non-invaded area. And this is even more clear if we look at the right side of the map. So these LDA scores, basically what they tell us is they highlight bacteria that are significantly more abundant in invaded areas, indicated in red, or in uninvaded areas, indicated in blue, irrespective of the presence of trees. And we can see that Opuntia stricta invasion leads to a significant increase in the abundance of up to 94 bacteria, while only 3 bacteria were significantly more abundant in uninvaded soils. So overall, wrapping up all the results of the soils, we found that native trees can create islands of fertility in the study area, meaning higher levels of humidity, nutrient content, and diversity of bacterial communities. Then among the 2 studied native trees, Bacillia nilotica caused a higher increase in all these metrics, especially in nutrients and so on. And this is already hinting, as I said before, that there is something there. Then on the other hand, Opuntia stricta also creates such islands of fertility. And I find this really interesting because it means that the presence of any tree or shrub in a savanna ecosystem, regardless of it being native or alien, or the growth form being a tree or a succulent shrub, can result in the formation of these fertility islands, which usually facilitate the establishment and growth of other plants. So I found it really interesting. But also interestingly, we found that Opuntia stricta affected soil bacterial communities more strongly than native trees. And the positive or negative consequences of these effects deserve further investigation. We didn't get to that yet. Now, the question is, how do these changes affect the establishment of Opuntia stricta? And as I said before, we did germination experiments with Opuntia seeds in the different studied soils. And we found that the percentage and speed of germination of Opuntia stricta were generally lower in invaded, on the right in red, than in uninvaded areas, indicated in green in this table. So these results basically suggest that perhaps the potential release of some allelochemicals by Opuntia stricta, which might retard its own germination. Maybe it's creating some negative soil feedbacks, aiming to avoid some potential intraspecific competition. But this is a hypothesis. Now, moreover, if we only look at the data that is highlighted in bold, these results suggest that the germination of Opuntia stricta might actually be facilitated by the presence of Bacillia nilotica trees, as we previously hypothesized. And such enhanced germination might be the result of the higher nutrient levels that we observed, as I showed you before, under the canopy of these trees. So these results might therefore explain this Opuntia bacilli association that we observed in the field, which is really clear in this picture, and this is why I'm showing it again. In the background of the picture, in the left, we can see these Opuntia patches established under the canopy of Bacillia and Nilotica trees, while we see no Opuntia plants that are growing under the canopy of this Spirostachys africana tree that we see on the front on the right, or in open areas with no vegetation. Sometimes you see it, but quite often you see more this pattern. Sorry. So now, Opuntia stricta, as I said before, is not the only invasive plant in Kruger National Park. Unfortunately, it would be really nice, but it's not. So in this figure over here, you can see the new records or the cumulative records of new alien, naturalized, and invasive species in Kruger National Park. So the new records of alien plants are indicated in green, the new records of naturalized plants are indicated in blue, and the new records of invasive plants are indicated in red. So as I said before, there are 146 alien plants already recorded in the park, of which some have already become naturalized and some are already invaded in the area. Now, among all of these species recorded in the park, there are 3 alien annual plants, which you can see in these pictures, which are Parthenium, Datura, and Xanthium. All of the 3 of them native to the Americas, and they are the most controversial taxa of the species in Kruger for management. And I will tell you now why. So the 3 of them are annual. And this means that during the growing season, which is the wet season in the park, they form extensive stands where they dominate absolutely the invaded communities. But after the growing season, when it becomes dry again, they die and they disappear. So managers think a little bit like, oh, they are there only half of the year. Why? This shouldn't be a big problem, right? Also, it is not known whether they have important negative impacts in the park. Therefore, managers at the moment, they don't know if they should prioritize these species for management or not. It's true that when you drive through the park in the wet season, you just see these species all over the place, but they are still not sure if they should prioritize them for management or not. The budget is not that big and they should really prioritize what is needed to manage. And besides this, these 3 species invade riparian habitats. And in arid areas, rivers playing an important role in the invasion of alien plants. The levels of water are constantly changing, and this leaves open areas that are susceptible for being invaded by alien plants. And these dynamics, they have never been really studied in detail in African savannas. So it's a really interesting topic. So what we did was to establish a project with the main objective of understanding the invasion and impact of these 3 species, Parthenium, Datura, and Xanthium, in Kruger National Park. And I must say, this is an ongoing project. We started it only a year and a half ago, more or less. So any ideas on how to improve it are very, very more than welcome. So first of all, we wanted to assess how these species come into Kruger. So here on the right, you can see the map of Kruger National Park. As I said, it has an extension of almost or around 20,000 square kilometers, more or less the size of Israel. And there are some main roads that get to the park. So what we did was to establish 4 transects in the 4 main roads that lead to the park. Each of the transects, it's around 20 kilometers, and it goes just along the road from the park to outside of the park. And in these transects, we are collecting presence and abscess data of these 3 species, so whether the species is there or not. And we survey these transects by car. But we also established transects along the rivers because we believe that they are the main pathway of introduction of these species in the park. They bring the seeds of the annual plants, and this is how they are mainly distributed, we believe. So we also established 1 transect in each of the main 6 rivers that are flowing through the park. And each transect goes from the river, once again, but this time 20 kilometers away from the river. And we survey these transects with aerial photographs that we take from light crafts, and each photograph has a 7-centimeter resolution. And so far, we took photographs in January, February 2022 and January, February 2023. We decided to take the pictures in January, February because it's the peak of the growing season, which is the peak of the wet season, and therefore is where we see more plant species. Now, with this, we expect to see that the presence of the invaders will be reduced with the distance to the river. And we are testing whether factors such as temperature and precipitation, river morphology, bedrock, and so on are affecting their distribution. Now, we hope to have the results of this study early next year. So unfortunately, I don't have results to show you yet on this, but I can show you how these photographs look like, at least, because we already have quite some photographs with us, so I can show you that. So this is one of the example of one of these aerial photographs. And what I didn't say before is that what we are doing is to use supervised learning models to identify these 3 target species in the pictures. And these models at the moment are being quite successful at detecting our target species. So in this picture, you can see 3 populations or 3 points of Xanthium, the presence of Xanthium. Here, it's a bit difficult to differentiate, but we have like 3 points of Datura and 1 point of Xanthium, or we also have some mixed populations of Datura and Xanthium. So it's been quite successful, but as I said, I'm sorry, I don't have the results yet. So I will move on to the next point. So importantly, we are also looking at the impacts that these species have in the field. Sorry. Now, to study impact, what we decided was to identify populations of these species through the park. Once again, you have the map of Kruger there on the left. As I said, it's really big, 20,000 square kilometers. Sorry for mentioning it again. But what you can see here is that we established populations of each of these 3 species. And mainly the populations of Parthenium are in the south, because are the main areas invaded by Parthenium. And then we have populations of Xanthium more or less in the center of the park, and populations of Datura in the north. I must say that the populations of Parthenium are really clear, because those areas are highly invaded by Parthenium. But the populations of Xanthium and Datura are really dynamic, because we found out during this project that sometimes during the wet season, so suddenly there is a flood, and then after the flood, there is a big invasion, let's say, of Xanthium. And then there is another flood that takes away Xanthium, and then Datura is growing. So there is this dynamic that within the same season, sometimes the areas are invaded by Xanthium, then invaded by Datura, and so on. But mainly, the ones in the center are invaded by Xanthium, and the ones in the north invaded by Datura. So what we did was to identify these 7 populations per species through Kruger National Park, and placed a pair of invaded and uninvaded plots in each of the populations. So in total, we have 42 plots of 7 populations per species, with 2 plots per population. Now, these pictures show you some examples of these per plots, how they look like. So A will be an area invaded by Datura, as well as C, and then B will be the per uninvaded area, and D will be the per uninvaded area of C. Then E will be an area invaded by Parthenium, F the per uninvaded area, and G an area invaded by Xanthium, and H the per uninvaded area. Now, we use these plots on the one hand to do vegetation surveys. So what we wanted to do was to study the impact of these 3 species on the diversity and composition of native plant communities. And here, in this slide, what you can see is 3 plots. The one on the left indicates the number of all plant species in areas uninvaded, indicated in gray, or invaded, indicated in orange, for the Datura populations, Parthenium populations, and Xanthium populations. So what we generally found is lower species richness, that you can see on the left, and diversity and lower diversity indexes, that you can see on the right, in invaded plots, with the strongest effect for Parthenium. So we saw an effect of in Parthenium invaded and invaded plots for all, while we found an effect of Datura in the diversity indexes, and we didn't find any effect for Xanthium. Now here you can see the results of ordination analysis comparing the species composition of invaded and uninvaded plots. And these results indicate that all 3 invaders modify the composition of native vegetation. So here we do find an effect of the 3 invasive plants. And finally, in the graph on the left, you can see that the number of native species clearly decreased with when you increase the cover of each of the invaders. And this is especially true for Parthenium invasions. However, on the right, you can see the number of alien species that are accompanying this invasive dominance, these 3 species. And there we didn't see any negative effect on the cover of alien species. It's quite an interesting result as well. So basically, all these results confirm that there is a negative impact of these 3 invasive plants on native plant diversity, with the most pronounced effect seen by Parthenium invasions. Now, besides this impact on plant communities, we are also studying the impact on soil physicochemical characteristics and microbial communities. Here in the top right, you can see a figure with the amount of precipitation and temperature through the year. So you can see that the top growing and wet season is January, as I mentioned before, December, January, February, and then March and April, it's already the end of the wet season and therefore the end of the growing season for these 3 species. So what we did to study these impacts on soils was that we collected in each of the 42 per plots that I mentioned before, we collected 5 subsamples from the top centimeters of the soil and then homogenized them into a single replicate. We use these mixed samples to measure pH, humidity, nutrients, enzymatic activities, the same as before as a proxy of the function of microbial communities, and then bacterial and fungal diversity and composition. We did it this year, both in January, which corresponds to this wet season, to the peak growing season. And then we also did it in March, beginning of April, which is when they start to die. So just to compare. Now, I don't have yet the results of the enzymatic activities or the bacterial and fungal diversity and composition because we are still processing that. But I can show you some really, really preliminary results. I only could analyze them yesterday for first time with really roughly for the pH, humidity, and nutrients. So here you can see the results for Parthenium in January, April, Datura January, April, Xanthium January, April, and basically it's just comparing invaded and non-invaded areas. And with these preliminary results, what you can see is that we didn't find a consistent impact of the 3 invaders in their soil characteristics, which is quite expected. But in accordance with the results of the plant communities, we saw that Parthenium seems to be causing the highest impacts. And these impacts are a little bit in the line with the islands of fertility that I talked about before for Opuntia invasion, so in which the soil nutrient levels seem to be higher in invaded areas than in non-invaded areas. But before we can properly discuss these results, we need to get the results from the other parts as well. So just something really preliminary. And even more preliminary, besides this, we are also looking at the impact on large herbivore communities, on both the presence and the diet of large herbivore communities. So in Kruger National Park, there are some of the most charismatic large herbivores, as you can see in these pictures. And to study the impacts on these communities, what we did was, on the one hand, we placed camera traps in each of the 42 per plots that I mentioned before through the year. So 7 populations per species in each of these populations per invaded and uninvaded plots. And in each of these plots, we place a camera. And these cameras take pictures every 5 minutes. And we get approximately 240 images per day, which is like around 45,000 pics per season. So what we are doing is to apply neural networks that read more or less approximately of the 80% of the images, and then the remaining pictures need to be inspected manually. Here you see some examples of these pictures of animals. I particularly like the one of the baboon with the hand up. And also one picture that I really like is this picture of 10 lions just lying there in the Datura invaded plot. So these pictures are going to tell us basically whether the animals avoid or not these dense patches. It has been seen before in other savannas that when you see, normally these areas are open areas, and suddenly you have a dense patch of vegetation, and then normally the herbivores tend to avoid these dense patches, especially something like the antelopes. So we want to see if something happens when there is this big invasion. But we cannot see much more than that. So to know if the herbivores are actually utilizing these plants, what we are also doing is that we are collecting in each of these 42 per plots, we are collecting dung of several common herbivore species like elephants, giraffes, impalas, water bugs. This is something that my daughters really like to see me collecting this food. And then what we do is to use DNA metabarcoding to find out whether the herbivores consume the alien plants and how large a contribution to their diet these 3 target invasive species might make. Once again, unfortunately, the results of the first batch of the that we collected in January and March will only be ready by the end of the month. I thought that they would be ready for this presentation, but unfortunately it was not the case. So I cannot show you any data yet. And then we will collect them again next year in January, just to have at least 2 years to compare. So overall, what we aim to see with this setup is the dynamics of the invasion of these 3 species through the rivers, from outside to the inside of the park, through the roads and over time, as well as their impact on the soil conditions and on the plant, soil microbial and large herbivore communities. And basically all of this has the ultimate goal of informing the park authorities how serious or not these invasions are and whether they are having impacts on the park. So they can decide at least whether or not they should prioritize their management. As I said, the budget is relatively limited, so they really need to prioritize to manage certain invasions. So we would like to tell them whether this species should be prioritized. So far, with these preliminary results that I showed you, it seems that Parthenium might have the highest impact. So probably they should prioritize the management of Parthenium, but we cannot say anything until we don't have the whole picture. So with this, I just wanted to say thank you very much for the invitation and for listening to me. Of course, this is not only my work at all, but the work of the whole team involved in this project, including my family, which are happy to come with me to this adventure. So big thanks to all of them too. And I will, of course, be really happy to answer any questions or discuss any aspect of the project with you. Thank you very much.
Thank you very much, Anna. What complex and complete work that you have done. Thanks again for explaining to us very smoothly.
Thank you.
Yeah, we have some questions already in the chat that I will read for you loud. First of all, we have a comment from Monse, a question. Was not Opuntia controlled in South Africa successfully, I guess?
Yeah, that's a good question. So Opuntia stricta is one of the highest success stories of management in South Africa and in Kruger. So at the beginning, when Opuntia became invasive and those thick infestations that I showed you, they started trying to control it with herbicides and cutting it. And that was like so much money and it was not successful whatsoever. Then they introduced one biological control that was not successful either. And then they introduced a second biological control that is extremely successful. The problem is that the populations went really down with the second biological control. And this biocontrol agent cannot jump that much from population to another. So what they have is they have some facilities in Kruger in which they have cladodes of Opuntia stricta and they grow there, the biological control. And then when there is a new population coming up, they have to release them. So they mainly manage it with that, but also when you find plants next to the road, because tourists tend to complain a lot because it's a really charismatic species. Many people know about it, about that invasion. So they have to cut it or use herbicides. So it's an ongoing, of course, with biological control, you don't get rid of it. It's ongoing and sometimes it becomes quite thick still. So for us, it was really interesting to see where can it go? So we know that we have to look in those areas to release the biological control agent, and also if it actually has any impact, because then, yeah, we should put more effort on that. Thank you. It's a really good question. And thank you for the invitation, Marcy. I cannot hear you. Sorry.
Sorry. We have another question from Irvin. Thank you for the amazing talk, Anna. I have 2 questions. The first one is relating to the baboons and the possible dispersal of Opuntia stricta. Do they have a higher presence for this fruit, or there is a native one that they like better?
Yeah, but baboons like a lot of things. They eat everything and anything. But the thing is that Opuntia stricta, normally in savannas you have fruits, but they are quite small. There is not much water or nutrients, so they are okay. But then you have suddenly this fruit that is like this, and it's really red and it's really sweet. So they go a bit crazy for it when they see it. that was also an interesting point that is interesting for me. I don't know if it's interesting for other people. The first biocontrol agent that was introduced was like, yeah, it's an insect and it's quite thick. Like the larvae are quite thick, and yeah. So what was happening is that they eat the cladles inside. So the baboons used to just take the cladles and eat all the insects as well. So yeah, this is why they really like Opuntia, because it's really fleshy and sweet.
Okay, I will read just the second question of Irving. The second question is regarding the patches of vegetation. Why do the large herbivores avoid them and prefer the open areas? I will expect that to not avoid them, since they could feed on that vegetation.
Yeah, but one of them, it's a good question actually, but one of the main things of the herbivores is to hide, especially the antelopes, because they need to hide from lions and leopards and so on. And the thing is that if they are in a dense patch, they cannot see far. So they cannot see if there is a lion approaching or a leopard approaching or something like that. So they don't really like that kind of habitats. They prefer if there is grass, but it's short enough that they can see far. And with some of these invasions, especially the Parthenium ones, they cannot see far. So that's a problem for them. So we expect that they are going to avoid these areas, which are the areas that they used to have access to the river and to drink water. So that can be problematic.
Although you can see the picture of the lions waiting there, no, just in case I have the lights coming.
Exactly.
Okay. We have a question from Francisco Javier Borrallo Vázquez. Thank you for the talk. It was very enlightening. I love the images. It really is a gorgeous place to work. Given the huge ability of these invasive species to shape the characteristics of the environment they invade, knowing little about the topic, if the impacts are positive, what could be the next steps?
I will assume they are not positive. Maybe I'm wrong. But for example, in savannas, you have the problem of encroachment, that when there are so many trees growing, then many of the species are replaced. And now that humans are managing quite some of these areas, there is a lot of encroachment, and that's a problem. So I will assume that even if they do these fertility islands, and at least Opuntia, it seems to be doing it like crazy, much more than the native trees, then they will be probably replacing native species, especially some, I don't know, some small annual plants and so on. And this is what we actually saw with the impacts on the plant communities, that they change a lot. And especially with Parthenium invasions, the different species, the diversity really goes down. So I doubt that they have a positive impact. It might be. If they do, then great. Then we can spend the money in managing something else. But I doubt it. I don't know. Maybe I'm being too negative. I don't know.
Yeah, this is what I like. It's surprised to be, no, like how Opuntia like increase the soil bacteria, for example, like more than like the native species, like the soil bacterial communities. But the thing is that it also makes an open door, no, for any other bacteria that might be pathogen for like even like wild tree species, and like it can, it's an open door for more invasions, so like for everything.
Yeah, on the one hand, it's an open door for other invasions. Sorry. Which can be a problem, and that's something that is commonly talked about in invasion science. But also something that I didn't mention is that in some of these invaded, non-invaded plots, we saw that there were 4 or 5 native plants that actually were really happy with invasions, and they grew there a lot. And that also, you can see it in dunes, for example. When you have an invasion, there are some opportunistic species that they really like those conditions and they thrive there. But those are normally really competitive plants that just displace the other plants. So there is a dynamic there that is a bit complicated and of concern.
Yeah, Francisco Javier also like comment now. So it's not usually a good idea to use an invasive species for, let's say, the ecological services that they might provide.
No, and that is being done. Like there is the example of Prosopis in Africa, in Eastern Africa, a huge disaster. In Vietnam, they are transforming the rice fields. This is a huge project to transform most of the rice fields in Vietnam for acacia plantations. And I don't know how they are going to get rid of that afterwards. And then you change completely the conditions of the soil. And yeah, it's not a good idea. There are many examples of that not being a good idea. In Galicia and north of Portugal with eucalypts. Not a good idea either.
True. We have some questions from Monse. Are the open areas, I guess that this is like from the first experiments that the first work that you presented, are the open areas just bare soil? What about grasses?
Grasses tend to, it's a good question. They tend to occupy these open areas. The picture I show, they look really open and bare soil, and it's because it was not the growing season. That was in May, I believe. That picture was taken, I think it was in May. So many of the grasses are not there that much anymore. But if you go in the peak growing season, most of the, not most, but a big part of the open areas are covered in grass. Yeah.
Also another question from Monse. These species may also be dispersed by mammals. It would be nice to relate the distribution of invasives and mammals movement. I guess it was related. It was related to the one that you say that they were kind of dispersed by the river. That was your hypothesis.
Yeah, I think for these three species, for the annuals, I don't think they are going to be dispersed by mammals that much, because I believe, we will see, but I believe Parthenium is dispersed by wind. Like each plant, I think it has like 2,000 seeds or something like that. And it just dispersed by wind, while the other two are mainly dispersed by water. But for Opuntia, it's definitely dispersed by animals. And we have seen examples of fruits eaten by birds and so on. So that's something I really wanted to have a look at. And here in Almeria, talking with someone from here, that did quite some work in Africa. Also, they told me that in other areas of Africa, not in South Africa, but they also told me that they saw with Opuntia that mammals eat it, but they don't disperse actually the seeds. They just poo, sorry for that, the seeds. And then it's ants that come quickly and they steal the seeds and they put them somewhere else. So I would really like to look at that, definitely. It's a really interesting part.
Yeah, I have a question regarding those. You say that I don't remember, like these three species that you presented. Are they annuals? You say, no?
Yeah.
Yeah, and but how do they rely on vegetative reproduction as well? Like I see like those photos of the patches that they create, and yeah, like it reminds me to all other clonal species that they are very invasive, and because like it's crazy how they reproduce and how they occupy like the environment. It's
from seed. It's really impressive, but it's really from seed. And actually you can, is what I mentioned before, that in these populations of Xanthium and Datura, for example, this year in January, part of the team went to do the field work. I didn't go there. And they sent us the pictures of the plots. They collected the samples and they sent us the picture of the plots. And then I was there 2 months later. And they sent me a picture of exactly this position. I had to look at the GPS many times. It was like thick with Datura. There was not a single Datura, and it was thick with Xanthium, the same plot. So it's just from seeds, and they grow on top of each other. And yeah, it's quite impressive.
It would be nice to see also, like, because I guess that whenever they die, so whenever it's coming, like the dry season comes, it really is like, how is the plant community after those ones they die, no? After like the invasive die. I guess that is just like bare soil. So it might be interesting also to see, okay, how in the dry season, the plant community, like the local one, is also affected by the invasion of those ones.
The thing is that they mostly invade areas that are quite bare soil in the non-wet season. And we did a study, I didn't present it here, we didn't publish it yet either, with Parthenium, and we saw areas invaded along the rivers, because Datura and Xanthium, they only invade the riverbeds so far, and maybe we find something else with the aerial photographs, we will see, but it's mainly around the rivers, the riverbeds. But Parthenium invades also acacia, Vachylia forests, used to be called acacia before. They also disturb areas around the park or around the the touristic campaments and so on. And we did a study in 5 different seasons to see how the soil was reacting to the presence or not presence of Parthenium. And we saw a little bit of impact in the soil in the growing season, but then after it dies, for the next 2 or 3 months, the impact was huge. And it's because suddenly you have all this organic matter decomposing, and then microbial communities, nutrients, the pH change. So it's quite interesting. But the thing is that the native plants don't germinate there until the next wet season. So yeah.
Yeah. I have a question of Irving. How should the dispersal of Opuntia by baboons should be addressed? Mostly because if they like to feed on it, I feel like it will be hard to eradicate if they are not removed almost all at once.
Yeah, there are several problems. One of them is that it's not feasible to remove all the plants and eliminate it from Kruger, because there are too many, and there are the small patches that you cannot really see normally, and you can walk through it, but it's not that easy. I got lost a couple of times, and you don't want to get lost in such an area. So it's not really feasible to get rid of it. Ideally, you release the biocontrol before they mature enough to have fruits. But also, even if you have a plant of this size, a small clado, you pass by and it gets stuck on your skin or on your clothes or whatever, and you take it to another part and it also disperses. And that we have seen elephants, they just carry it. Like it's not so easy to stop the dispersal by mammals. Yeah, that's a good question.
We have some comments. Murica Peya says, thanks for the nice presentation, Anna. Also like some congrats coming from Mylene Mariette. Fascinating. Thank you. Another question. Just a final question from Francisco Javier Borrallo. He says, out of curiosity, are those examples of using the potential of those invasive species for financial exploitation or an honest effort for ecological restoration?
The invasive species.
Are those examples of using the potential of those invasive species for financial exploitation? I mean, I guess he refers that those ones that are invasive, if they are used also like for financial exploitation. So if whether they are used, they have any economic value, I guess.
Mainly
or an enormous effort for ecology. Yeah.
Mainly Opuntias too, especially Opuntia ficus indica, but also Opuntia stricta in Eastern Africa is used for the fruits and to be used for feed cattle and other domestic animals. There is lots of controversy with Opuntias, that for sure, like big controversy. I was working on that during my postdoc, and I'm still involved on that because I find it fascinating. And it's because you suddenly have an arid area where there is not that much production of other species, and then Opuntias can grow everywhere. But then people start using it, but then they don't want it anymore because they realize they take over and they have so many negative impacts, even in health, of human health and so on. And then getting rid of it is really difficult. So it's, for Opuntias, there is a lot of that. For the others, really little. For example, Parthenium was introduced in India and it took over, and it has some chemicals that create really bad skin problems in humans, and there is, like, people don't want it. Datura and Xanthium, they are also a bit poisonous, so it's not something that it's really wanted.
Another comment from Monse. With regard to cover, it seems that they are not invaded or highly invaded areas. No, sorry. It seems that there are no invaded or highly invaded areas, but not medium invaded areas. I think she refers to those graphs that, yeah.
Yeah, I believe that it's, so you have the river flowing, and in these areas where the water starts getting a bit slow, then the seeds get stuck there, and then and then where the river keeps flowing, and so in the moment that the seeds arrive, they just take over. So it's really difficult to just see one plant here and there. It's normally thick infestation. And with Opuntia, the same. If you leave it there for a little bit,
Okay, Francisco Javier is commenting on his question. He says, I meant for the use of eucalyptus on Galicia or the transformation of rice fields of Vietnam, if they are an effort of restoration or is just greenwashing of financial exploitation of the land.
So
but I guess that is not the case in there. No?
Eucalypts were introduced for making paper. And there is a big industry of that. There is in Pontevedra, actually, where I come from. And this is the reason why I studied innovation science, because I grew up seeing these eucalypts. And that was the main reason. Then people started to realize that it's really difficult to get rid of them. And also they changed completely the soil, and the microbial communities with eucalypts is quite impressive. They cannot disperse so much. So this is why there is a bit of controversy. But in the areas where they are growing, they have huge impacts. So slowly people start realizing there are people that still get lots of money from it, and then they are happy to have them, and other part of the population that they realize it's a problem. In Vietnam, it's really new that they are doing that. So people didn't realize about the negative impacts yet. So culturally, quite often there is like, oh, this is really good, this gives me money, or it's really pretty, and then, oh, it has negative impacts. Sometimes the people don't realize about the negative impacts, sometimes they do. So sometimes when they don't, the species are controversial because some people don't like it, some people do. When they have really strong negative impacts, as Parthenium in India, nobody wants it anymore. So it depends on the case a lot. Yeah.
Okay, thank you very much for those answers. I have just one. I'm not very like I don't know much about like invasive ecology, but like I kind of heard that there is kind of like a time lag between when it gets introduced species and then when it gets invaded. How about like those species that you talk about? Like how was Opuntia, like the case of Opuntia or those animals were very fast. They had also this time lag to be very invasive or not?
I know more, like Opuntia, as I said, was introduced in 1953. And by 1998, it was already absolutely heavily invading Kruger National Park. Parthenium, if I'm not mistaken, it was introduced in 2003, I think, to Kruger. And a couple of years later, it was already taking over. So it really depends on the species. Some of them have a big time lag, some of them don't, or lag phase, some of them not. Yeah, I don't, of Datura and Xanthium, I know they were first introduced, I think, in the 50s in Kruger, 50s, 52, 54, I think. I'm not 100% sure when they started to take over. That I'm not 100% sure. I think it might have been a decade later or something like that, but I might be totally wrong. It's just from the top of my head.
Sorry again. First online seminar.
Sorry for that.
No problem. Yeah, thanks again, Anna, for coming. I think that's a wrap. We're almost start. Thanks again for coming, and thanks all the audience in YouTube. And see you next time.
Thank you. Thanks a lot.